Hello viewers and my dear students. I'll be taking today the video I'm preparing or taking a class on DNA structure, and I like all of you to view this video, like, share and subscribe my channel. And to start with now, first we'll start with what is a DNA. DNA stands for deoxyribonucleic acid. It stands for deoxyribonucleic acid. And the length or the size or length of DNA is measured in terms of number of nitrogen bases present. In terms of number of nitrogen bases present. And so here we can calculate the length based on the number of nucleotides present. Like for example, the nucleotides present in case of E. coli, that is in bacteria, in E. coli, they are present as 4.6 into 10 to the power of 6 base pairs. 4.6 into 10 to the power of 6 base pairs. Whereas in a haploid human cell, it is about 3.3 into 10 to the power of 9 base pair. That is in a haploid human cell. And then in a bacteriophage, that is 5x174. This is a bacteriophage. A bacteriophage is it is a virus which infects bacteria. So in a bacteriophage, it consists of only 5,386 bases. It is not in pairs, it is in bases. So it means that here it is single-stranded DNA. Here it is single-stranded DNA. And so wherever there are base pairs present, we can call it as here it is a double-stranded, and wherever there are only bases present, it is called as a single-stranded DNA. So this is how the length can be measured. Now next we will be doing about the few historical studies, few historical events or studies made in the field of DNA. So it is a brief history in the field of DNA. So first, the nucleic acids were first discovered by Friedrich Miescher. Nucleic acids were first discovered by Friedrich Miescher, and he called it as nuclein. He called it as nuclein. This discovery he made in the pus cells of an owl. He made this discovery in the pus cells of an owl. And later, the term nucleic acid was given by Altman. Altman coined the term nucleic acid, and various other studies are also made in order to understand the structure of DNA. The most other important studies which are made to understand the structure of DNA is another study by Wilkins and Franklin. They used the technique X-ray crystallography. They used the technique called X-ray crystallographic technique. So by using X-ray crystallographic technique, they observed that DNA is a right-handed helix. By their study, they observed that DNA is a right-handed helix. Helix means it is a twisted structure. So it is a right-handed helix. And then other study by Pauling. Pauling study is he observed the presence of hydrogen bonds. Presence of hydrogen bonds between the two strands. DNA is a double-stranded molecule. There are two strands of DNA. So the presence of hydrogen bonds. Hydrogen bonds are present between the two strands. So presence of hydrogen bonds was given by Pauling. So based on these studies, finally, Watson and Crick, they described the structure of DNA, which is called as a double helical model of DNA. So final structure of DNA was given by Watson and Crick in 1953. Watson and Crick proposed this structure, explained this structure in 1953 by considering all the other previous studies. So, according to Watson and Crick, DNA shows a double helical structure. It shows a double helical structure. That means it consists of two polynucleotide strands. Two polynucleotide strands. Double helical structure is it consists of two polynucleotide strands, and these two strands are twisted around each other. So, structure he described it as twisted ladder-like structure. He described the structure as a twisted ladder-like structure, which consists of two polynucleotide strands. So now let us see what are the structural components and what is a polynucleotide. So here we find, when we see the double helical structure, each one is a strand which is described as a ribbon-like structure. Each one is a strand. So these are the two strands present and they are twisted. So its structure is compared as a twisted ladder-like structure. So now, what is a nucleotide? We will see what is actually nucleotide. We are calling it as a polynucleotide strand. So we should see first what is a nucleotide. Nucleotides are considered as they are structural units of nucleic acids. They are structural units of nucleic acid. So here we are focusing on first the DNA structure. Once we discuss DNA structure, we can compare it with RNA also. So nucleotide is the structural unit of nucleic acid. Now what is a nucleotide and what does it consist of? A nucleotide is composed of three components. A nucleotide is composed of three components. They are called as nitrogen bases. First component is called as nitrogen base. The second component is called as a sugar. And the third component is called as a phosphate. So these three together, that is nitrogen base, sugar and phosphate, these three together, they form what is called as a nucleotide. And nucleotide forms the structural component of nucleic acids. Now we will see one by one what is a nitrogen base, how many types are there, and what is a sugar and what is a phosphate, before actually going through to the exact structure of the DNA. So first, now I will continue with what is a nitrogen base. First, we will see with what are different nitrogen bases and what are all, how many are present in DNA. So nitrogen bases, if we take nitrogen bases, there are 4 types of nitrogen bases. There are 4 types of nitrogen bases present in DNA. So 4 types are, first one is called as a adenine, the second is called as a guanine, and then cytosine, the third one is called as a cytosine, and the fourth is called as a thymine. So these are the 4 nitrogen bases: adenine, guanine, cytosine and thymine. So of these, the first 2, adenine and guanine, the first 2 are called as purines. So purines are adenine and guanine. These are called as a purines. And other features of purines are, these are double ring compounds. They consist of double ring, or double ring structures are also called as dicyclic structures. Double ring structures are also called as a dicyclic structures, and they are heterocyclic. They are dicyclic and heterocyclic. That is, they consist of 2 rings. 2 rings are present. 2 rings are present, and these consist of nitrogen other than carbon. Other than carbon, if they have nitrogen also, other than carbon, if they have nitrogen also, then they are called as heterocyclic. So in purines, they along with it, carbon, nitrogen also are present. So these adenine and guanine are called as a double ring compound or dicyclic. And other compounds, other 2 are called as pyrimidines. Pyrimidines are thymine and cytosine. Thymine and cytosine, these are called as pyrimidines. And these are single ring compounds. Single ring, or they are called as monocyclic. These are called as single ring, or they are called as monocyclic compounds. And these are also, just like purines, these are also heterocyclic. Just like this, these are also called as heterocyclic compounds. So now these purines and pyrimidines, they bind with each other. So nitrogen, in nitrogen bases we find the one purine always binds with a pyrimidine. So now let us see. So these are the components. Now let us see the second component, that is the sugar. Second component, that is the sugar. So sugar present in DNA, it is called as a deoxyribose sugar. Sugar present is called as a deoxyribose sugar. This deoxyribose sugar, it is a pentose sugar. That is with 5 carbon atoms. It is a pentose sugar with 5 carbon atoms. And this sugar binds to phosphate. That is third component. Sugar binds to phosphate. And the bond between sugar and phosphate is the ester bond. The bond present between sugar and phosphate, it is called as a ester bond. So sugar has 5 carbon atoms. So this ester bond is formed between third and also fifth. That is, third carbon atom of the sugar, one sugar, is connected to fifth carbon atom of the next sugar. So here I have shown the sugar has 5 carbons, 1, 2, 3, 4 and fifth carbon, and third carbon is attached to fifth carbon of next sugar with the help of a phosphate. And this bond is called as between sugar and phosphate. It is called as a ester bond. And because one phosphate is connecting the two sugars of the two different nucleotides, therefore this is also called as a phosphodiester bond. Phosphodiester bond. So a phosphodiester bond is formed between sugar of the previous nucleotide and sugar of the next nucleotide. So when we observe this, so how we can? So here if we take, say this is adenine is present, suppose this is here cytosine. So here another sugar is present and these two sugars again are connected by a phosphate. So this bond which is connecting the two sugars with the phosphate, this is called as a phosphodiester bond. Phosphodiester bond. So that is the sugars present. Now next we will see what is the phosphate. How phosphate is present. So phosphate is present in the form of phosphoric acid, that is PO4 phosphate, or it is present in the form of phosphoric acid. Because it is present in the form of phosphoric acid, it makes the DNA molecule acidic. DNA molecule will be more acidic. And this phosphate is attached to the sugar. Already we know sugar and phosphate together, they form a bond. So this phosphate is attached to the sugar with the help of the ester bond. Now all three together, now they are forming its unit of nucleic acid or DNA, that is called as a nucleotide. So nucleotide has nitrogen base, sugar and phosphate. Now let us see the nucleotide of each nitrogen base. We know there are 4 nitrogen bases in the DNA. So first if we take adenine, it is attached to sugar. When any nitrogen base is attached to sugar, then it is called as a nucleoside. So nitrogen base plus sugar is called as a nucleoside. So adenine plus sugar, its nucleoside is called as adenosine. Its nucleoside is called as a adenosine. Now adenosine when attached to phosphate, it becomes a nucleotide. So nucleotide of adenosine is called as adenylic acid. It is called as a adenylic acid, or it is also called adenosine. Adenosine monophosphate. It is also called as adenosine monophosphate, or it is called as adenylic acid. This is the nucleoside and nucleotide of adenine. Similarly, guanine plus sugar, its nucleoside is called as guanosine plus phosphate. When phosphate is added, it becomes nucleotide. So it is called as guanylic acid or guanosine monophosphate, GMP, AMP or GMP. Adenosine monophosphate, AMP, or guanosine monophosphate, GMP. Similarly, the other two, cytosine plus sugar, it is called as cytidine plus phosphate, that becomes a nucleotide. It is called as cytidylic acid or cytidine monophosphate, cytidylic acid, or it is called as a cytidine monophosphate. Similarly for thymine plus sugar, its nucleoside is called as a thymidine. Its nucleoside is called as a thymidine plus phosphate. So plus phosphate, when it is added, it is called as thymidyllic acid. Thymidyllic acid. It's called as a thymidyllic acid or thymidine monophosphate, or it is called as a thymidine monophosphate. So these are the different nucleotides and nucleosides of the 4 nitrogen bases. Now let us see what is the actual structural feature of a DNA. So let us see what is the structural properties or structural features of a DNA. A DNA, already we just discussed, it is considered as a twisted ladder-like structure. So it has 2 strands which are twisted around each other, and the 2 strands appear as ribbon-like structures. So this twisted ladder or ribbon-like structures, twisted ladder-like, and the 2 ribbon-like structures which are actually made up of sugar and phosphate. These 2 ribbon-like structures, they are made up of sugar and phosphate, and this forms the backbone of DNA molecule. Backbone, that means it gives that structure to DNA. Backbone is which gives strength or structure to DNA. So this is the sugar phosphate backbone. So if we understand that diagram in another way, this is how we can diagrammatically we can represent the 2 strands, ribbon. How are the 2 strands made up of? They are made up of sugar and phosphate. Sugar and phosphate. So this is how we can. So this sugar is attached to a nitrogen base here, which is attached to another, the 2 nitrogen bases, and then to the sugar this side. Similarly, here the nitrogen base and the sugar. And these 2 sugars of these, they are attached to the phosphate. So here sugars of the 2. This is the sugar of this. This is attached to the phosphate. Sugar of these 2 will be attached to a phosphate. So this is called as this sugar phosphate structure, sugar phosphate, that is called as a backbone of DNA molecule. So actual strands are, these strands are made up of sugar and phosphate, and the 2 strands are connected by these nitrogen bases. Now the 2 important features of the DNA is, first important feature is the anti-parallel nature. Anti-parallel nature of the DNA helix. Anti-parallel nature is the 2 strands are opposite in direction. The 2 strands are opposite in direction or polarity. It is also called as polarity. Opposite in direction or polarity. So we always give the 2 strands as, suppose the 1 strand, this is in 5 prime, the direction we measure as 5 prime, the other end we should give it as 3 prime. Similarly, this side if it is 3 prime, the other end we give it as a 5 prime. So we denote the direction of the strands as, suppose one is in this direction 5 prime to 3 prime, other one will be 3 prime to 5 prime. So this 3 prime and 5 prime, how we denote is, we can understand in this diagram clearly. That is, this end where the 5th carbon is attached to phosphate, this is called as a 5 prime end. The other end we find the 3rd carbon, here it is the 5th carbon which is attached to phosphate, so it is named as 5 prime. The other end is the 3rd carbon which consists of OH group. It is a free carbon, 3rd carbon attached to OH group. This is called as a 3 prime. So suppose this is the direction, if we give 3 prime to 5 prime. The same thing opposite we find on the other strand. That is here the third carbon atom is free. It has OH group. So this end is 3 prime end. The other opposite we find fifth carbon attached to phosphate. So this end we call it as a 5 prime end. So here if we take this will be in this direction. So the two strands always run in opposite direction. They have opposite polarity. So that feature is called as anti-parallel. The second important feature of DNA is the complementary nature. Second important feature, it is called as a complementary nature. So complementary nature of the two of the DNA molecule is the base pairing. Base pairing is called as a complementary nature. That decides what is the meaning of complementary. So base pairing is always a purine always binds with a pyrimidine. Purine always binds with pyrimidine. Suppose there is a purine on one strand, there will be pyrimidine on the other strand. Purine always we find. So suppose this is a adenine. We know purines are adenine and guanine, and pyrimidines are thymine and cytosine. Suppose this is adenine, always binds with thymine, and the bond present between nitrogen bases is the hydrogen bonds, and adenine and thymine always bind with the help of two hydrogen bonds. Similar, so this is a purine and thymine is a pyrimidine. Similarly, suppose if we take another purine, say guanine, binds with cytosine, pyrimidine. So between guanine and cytosine, there are three hydrogen bonds. That is called as complementary. That means always a purine binds with a pyrimidine. So if on one strand if there is a purine, we find its opposite strand, other strand, it will be a pyrimidine. So if we know the sequence of one strand, we can easily know the sequence present on the other strand. So suppose here we find some sequence of nitrogen bases on one strand, easily we can identify the sequence of nitrogen bases on the other strand. So this feature, this is called as a complementary nature. So these are the two important features, antiparallel nature and the complementary nature. Now, next other structural aspect which we can find is, already discussed that DNA helix shows a twisted ladder-like structure. So when it is showing a twisting, it is normally a right-handed helix. It like right-handed means clockwise. It shows a clockwise turning right-handed helix. So when it shows a clockwise turning, it twists in turns in such a way that it leaves a small gap in between, which is called as a minor groove, and it also leaves a large gap. Suppose this side if you also, this is a small gap, this is a large gap. So small gap is called as a minor groove and large gap is called as a major groove. What is its importance we will be discussing in the coming lectures, which I am going to present. What is the importance of this minor and major groove. So that is what is a important structural feature. So it has one major and the one minor groove. Now next we will see what are the dimensions of DNA molecule. Measurements. Dimensions means what are all the measurements of DNA molecule. Let us see the dimensions of DNA molecule. So dimensions if we take, first we will see what is the length of one turn and what is the meaning of one turn here. The meaning of one turn here, as I have labeled, we should start with minor groove here and come up to another starting of minor groove. That means we should include one major and one minor groove. That is called as one turn or one coil. So first important is one turn or one coil of DNA has a length of, the length I have measured given here. What is the length here? Its length is 3.4 nanometers, or in terms of angstroms it is 34 angstroms. DNA is a very, very large molecule. We cannot give the entire length of DNA of any individual. So we take only a unit length. We have to take only a unit length. That is, say here if we take only in terms of 1 turn. Or we can give the length in terms of base pairs also, or number of nucleotides present or number of base pairs present. That we are going to see now. So suppose there is a 1 turn or 1 coil. So what is its length in terms of nanometers? It is 34 angstroms, or it is the 3.4 nanometers. Now in this 1 turn we find 1 turn consists of 10 base pairs. There are 10 base pairs present. Base pair means already we discussed adenine and thymine form one base pair. Similarly, guanine and cytosine form another base pair. In this way, if we count a total of 10 from here to here, there will be 10 base pairs present. So, in 1 turn, there will be 10 base pairs. And what is the length of 1 turn? It is 3.4 nanometers or 34 angstroms. So, therefore, the next dimension, distance between two base pairs, two adjacent base pairs. Suppose we observe these two adjacent. These are the two adjacent base pairs. So distance between, this is the two adjacent base pairs. So distance between two adjacent base pairs, in terms of nanometers, if we take 0.34 nanometers, or in terms of angstroms, it should be 3.4 angstroms. 0.34 or 3.4 angstrom. That is the distance between the two adjacent base pairs. Therefore, in 1 turn, it fits about 10 base pairs. Now, next dimension is the total angle of 1 turn or 1 spiral. Turn is also called as a spiral. It is also called as a coil or a spiral or 1 turn. What is the total angle? Angle means how much it turns, what is the pitch, or how much it turns. So the total angle of 1 turn is always 360 degrees, the total angle. So we have to imagine an axis here. On an imaginary axis, if we observe, it takes a complete turn. So here, suppose this is an imaginary axis, you can observe from here to one, I told you, one minor groove coming here, then reaching another one minor groove. So if we take, it will come to 360 degrees. So therefore, the next dimension we can give is angle between two base pairs. Just like we have given a distance between two base pairs. Similarly, the angle, how much it turns, what is the angle between two base pairs? It comes to 36 degrees. Comes to 36 degrees. The next aspect, the next dimension which we can give is the distance between the two strands. Distance between the two strands. Distance between the two strands. Distance between two strands is measured. We can give in terms of nanometers. If we give, it will be 2 nanometers, or in terms of angstroms, we can give it as 20 angstroms in terms of angstroms. So that is the distance between the two. So these are what are all the different measurements or dimensions. So now the last aspect, last structural aspect which we will be considering now is what is called as a, we will consider what is called as, we call it as a Chargaff's rule. Repeat again, what is called as Chargaff's rule. Chargaff's rule. This rule was given by the scientist Erwin Chargaff. Erwin Chargaff. So Erwin Chargaff observed that always the purines and pyrimidines, because DNA is a double-stranded molecule. It consists of two polynucleotide strands. So DNA is a double-stranded molecule. So According to Irving Chargaff, always in a double-stranded DNA molecule, purine to pyrimidine ratio always is 1. That means purines are equal to number of pyrimidines. So the ratio is 1:1. Purine to pyrimidine ratio is 1:1. So this is always we find according to Irving Chargaff. Always if there are purine on one strand, we find pyrimidine on other strand. So how we can write it here as A plus G is equal to T plus C, where A and G are purines and T and C are pyrimidines. So this is called as a Chargaff's rule. Based on this, there will be some questions also which we will be discussing in my other coming videos. I will be discussing at the end of the DNA structure. I will be discussing about the different types of questions which we can expect in a competitive exam, any other exam, medical entrance exam. So, this A plus G and T plus C. A plus G is equal to T plus C. This is called as Chargaff's rule. This we can write it also as A plus G by T plus C equal to 1. So, both the ways we can write. This is what is called as a Chargaff's rule. Another thing which we have to know is what is called as a base ratio. Base ratio. This is different from Chargaff's rule. Chargaff's rule is applicable to all the DNA molecules of all organisms wherever DNA is a double-stranded molecule. Except few, one example earlier we discussed is 5x174. Otherwise, in general, DNA is always a double-stranded molecule. So, wherever DNA is double-stranded molecule, this Chargaff's rule will apply. Base ratio is the ratio of the bases, that is base pair. We know A and T forms one base pair. Similarly, G and C forms another base pair. So, A plus T by G plus C equal to 1. This is called as a base ratio. We find there is a difference. This is purine to pyrimidine. But here, based on their how they bind, A plus T, that is AT ratio and GC ratio. So, this is always equal to 1. This may be different from one species. This is actually different from species to species to species. This is different from species to species. Like for example, in humans, it is about 1.93. It comes in, whereas in E. coli, it comes to about 0.96. So it varies from one species to the other. So with this I just conclude mine for this video. My coming videos I will be discussing continuation of this topic. Please like, share and subscribe. Thank you very much.